Thank you, and thank you for asking me to give this talk. Network anatomy, really anatomy. You hear all the time that we study connectomics, but I think that sells it short, that this is our projects that we've been working on for now 7 years here and 10 years before, are really about complete anatomy, going from cell biology to neuronal morphology to connectivity and all of the ramifications that come from that. And I don't want to concentrate, actually, on the functional connectomics, functional anatomy, but really concentrate on the next decade, which I really think the next decade is the decade for cell types and connectivity and cell types and anatomy. So I have an outline, wow. An introduction that I'm going to try to go through fairly quickly. It's mostly historical, the evolution of our thinking about connectomics, and introducing the idea of CAP, which I'll get to in the next slide. Then cell types and connectivity, cell types and morphology, a new research program that has been sort of a glimmer in the back of my mind for 5-6 years now, but it's really happening finally. And then ending with some big questions that we're poised to answer really right now, but over the coming years. So first, the introduction, again, that I'll try to go through fairly quickly because it's largely historical. So where does CAP come from? It comes from complete, accurate, and permanent science. And this is from talks of Sidney Brenner. The first time I could find that it was written down is a summary of a talk in 2003 at Columbia, where he discussed complete, accurate, and permanent data types. And he really, in this talk, and most of his talks historically on this topic, it was about sequencing, about things that you sequence, you get a letter, that's not going to change. That's ground truth, plus or minus. No ad-libbing. But thank you for laughing to remind me not to ad-lib too much. Okay. I was lucky enough to start doing connectomics in a fabulous environment that included Jeff Lichtman and Sebastian Sung at Harvard and MIT, when we all decided in 2006, roughly simultaneously, that this is what we want to do. And we taught the first connectomics course, I'm pretty sure, EM connectomics course at Harvard and MIT in 2007. And fantastically, the first lecture was Sidney Brenner. And this is Sidney in the middle at dinner with Sebastian, Mehmet Yannick, who some of you might know, Xiaowei Zhuang, and the early middle-aged me. I took notes. I have the same bad handwriting that I've always had. Sidney Brenner, 070918 918 of 2007, and his lecture started with CAP, complete principle, complete, accurate, and permanent. Only science that has this is sequencing. And he went on to talk about his great work with White on the connectomics, the anatomy of C. elegans, and said, this is almost CAP, and he had some caveats, but he was hinting strongly that at some point this kind of EM connectomics might become a CAP science. The course was called Connectomics. Jeff Lichtman gave the introduction, his dictionary definition of connectomics. And I was sitting next to Sidney and saying, do we want to call it connectomics? What about microanatomy? Shook his head. Nanoanatomy? Really shook his head. He grabbed my pen and said, anatomics. He didn't say anything, but we were politely listening. But he wrote down anatomics. Well, perhaps, but let's call it anatomy. Let's call it what it is. I've shown this a few times in the past several months, but it's something that made me happy. Along with a large number of others who are my age in neuroscience, many of us read this 1979 book, or Scientific American, that came out in roughly 81 is a book. I read it in 1982, and there are wonderful articles by Hubel and Wiesel, et cetera. But at the very end, there was an article by Francis Crick, which I know I read and filed away somewhere. And he talked about all sorts of great techniques, but it said some things are impossible. So let's think of techniques that are almost as good as the impossible. And in this paragraph, he said, well, maybe we can trace circuits with rabies, because it would be impossible. It's no use asking for the impossible, such as, say, the exact wiring diagram for a cubic millimeter of brain tissue and the way all of its neurons are firing. So somewhere in the back of my head, I said, well, maybe impossible. And here are a bunch of slides from more than a decade ago that I'll go through very quickly. This was our way of thinking. How to reverse engineer a computer. How would you do that? Well, you first look at what it's doing. And in the earliest computers, such as Colossus, that saw the Enigma codes with Alan Turing and others, you could see what the computer was doing by seeing the flashing lights. And on the orange lights are essentially counting to 10 in various ways, doing decimal arithmetic. How would you figure that out? Well, if you build something, you have a wiring diagram, and here is a decimal arithmetic unit. So could we do this in the brain? First step, sort of started around 2000 in my group. We said, gee, let's do calcium imaging in the visual system. So we did that for a number of years, and it was fun. It continues going now. But really the idea was, this is an anatomical method to see all the neurons active. Anatomy is somehow related to connectivity. And all my past research had been about activity and connectivity, but without the anatomy. Wouldn't it be great to add the anatomy? And at first we thought viruses or something might be the ticket, but then Winfried Denk came out with his amazing sort of groundbreaking article on the early high throughput serial section EM, and all of us believed that let's do serial section electron microscopy, but our group wanted to do it in the context of physiology. Jump forward a decade, I guess, we continue to do all of our mouse connectomics in the context of physiology, but now we're doing the physiology in the cubic millimeter, either at Baylor or here at the Allen Institute, with next-gen 2-photon, 3-photon genetically encoded indicators. Let's look at what the computer does and then look how it's wired up. And it was a very computational framework that all of us thought in up to today, but you'll see how that's changing. I don't have a single picture of a microscope. I don't have a single, I'm not going to say tera, peta, exa. None of that. This is all about data and analysis. We have three data sets that you've heard about: Pinky, Basal, and Mini Mouse, the mice that we've scaled up. The team has scaled up from modest 500 neurons to slightly immodest 4,000 neurons, and then the grand data set, Mini, that's beginning to be analyzed, the whole cubic millimeter, the impossible cubic millimeter with 100,000 neurons and 1 billion synapses. The only reason I want to show a bit of technique is not to show the technique, but let me say, just for people who haven't heard me yammer on a million times, in these experiments, you first start with calcium imaging, then you go to sample preparation for EM, sectioning, EM imaging, co-registration of the two data sets, and then finally segmentation. But I really just want to call out the amazing team that that's what Showcase is about. But I just feel we all feel tremendously lucky to have this amazing team from Joanne sectioning, Dan and Wenjing imaging, Gayatri and team, these are all teams too, stitching and aligning, analysis, Agnes, Forrest, Casey, Sharmi, and many of the others. The fantastic leadership team, Nunu, Shelby, and our department, and my own personal leadership team, Cheryl. And finally, just an amazing group of people who I hope most of you know, Mark, Derek, Russell, Sam, and Layla, who do everything from co-registration through lots of computer work, et cetera, et cetera. Enough introduction. What are the big questions? Why do we do this? What next? Well, first, anatomy, morphology. What are the morphological cell types, especially using ultrastructural, cell biological features such as nuclear morphology, organomorphology, and location? I have an asterisk for all the studies that, all the types of data that feed into the chandelier story that you heard yesterday. Connectomics, what are the determinants of connectivity? There's topographic specificity. You heard yesterday, if a neuron isn't in the neighborhood, it can't be connected. That's important to know and use as a control. Emphasis, people can change, people can learn. And I have certainly over the past 7 years learned from Ed and Han Kui and all the amazing people in cell types. That cell types, that's the thing. And that's, if you ignore cell types, you ignore the heart of neurobiology. And just for connectomics, do certain cell types tend to connect to or avoid each other? That we have that in the chandelier story. Sort of the old way of thinking, last decade and this decade way of thinking are functional connectomics. What is the relationship between function and connectivity? But also new things that we're just beginning to explore, such as network structure, random small worlds networks, low-dimensional motifs, et cetera, et cetera. And then, for lack of a better word, morphoconnectomics, but really morphological anatomy. What are the morphological correlates of synaptic weight, spine volume, PSD area, synapse location, biophysical integration, et cetera, et cetera. But again, what did we all learn over the past number of years? That the morphologies of genetically defined cell types that have their unique morphologies, and here, and if people don't know this figure, the various cell types and the various examples of cell types, we can use that as a Rosetta Stone to bring our connectomics or our anatomy into perspective. So in the next section, I'm going to talk about current and ongoing work on cell types and connectivity, and going from just the data to, for lack of a better word, discovery about different types of cells and how they interrelate with each other. You have seen slides like this, but it's just important to start with an example, one of the early examples of a cell that we looked at in quite some detail, a basket cell that really for the first time, and people can look at 4,000 synapses onto a cell and almost 2,000 synapses out of that cell. What's the logic of this, and how does it relate to cell types? And we have that through many different types of cells, basket cells, chandelier cells, the various forms of VIP and somatostatin cells. And this is a slide just to show how far we've gone. A little bit more than a year ago, well, this is really the work of Nunu and Agnes, began looking at these input-output diagrams and identifying the different classes of inputs to cells, the excitatory and inhibitory, but then the basket cell inputs, the chandelier cell inputs, and the somatostatin inputs, really getting at the W alpha beta with great confidence on the numbers. And I'm not going to show that again. By quantifying it, the numbers of the different types of inputs to pyramidal cells and other targets, they were able to get their first real results in this cell type connectivity space. Not quite enough to publish an encyclopedic paper, but enough to know that there's pattern there. And this first study was on the bottom 7 different types of axons, or 6 different types of axons. The basket cells were first all of them, and then subcategorized. Basket cells, somatostatin, VIP cell 1 and 2, and then chandelier cells. And their targets, which are in excitatory cells, apical dendrites, basal tufts, and various types of inhibitory cells. It's a complicated slide, but let me just point out 2 things. One, chandelier cells only contact one thing, the green axons. And for instance, there's great specificity that jumped out. And this is sort of a new take on an old story, that VIP1 cells, this subclass of VIP cells, VIP2 cells, the subclass of VIP cells, is extraordinarily targeted towards inhibitory dendrites. There is a mixed up story that it was sort of 50-50 excitatory inhibitory, but VIP1 is sort of like other cells in the targeting, but VIP2 cells target inhibitory axons with great strength and fidelity. A sea change happened when we got a new analysis infrastructure, and all of the studies you saw yesterday, posters, and that I'm going to run through now, came from an analysis infrastructure that allow us now, cell type by cell type, pathway by pathway, to study things such as the beautiful third time of this movie, the chandelier cell inputs to all of the local pyramidal cells in a part of cortex, or a number of wonderful studies that are slightly under the radar, although you saw a movie yesterday of cell types in morphology. These beautiful work by Forrest and Layla about soma shape and I'm going to skip to the next slide, soma shape and size. For instance, and if you look at nuclear size in the middle plot, the nucleus volume, as everyone knows, changes in depth. But if you do slightly in the right-hand side, you look at a measure of nuclear morphology. I won't tell you exactly what it is, that there's a great heterogeneity of this nuclear morphologies in layer 5, but it's much better another one of Forrest's lovely movies. You have 3 classes of layer 5 pyramidal cells on the left with different nuclear morphologies clearly, and then this summarizes the whole game, that they go from nuclear morphologies to cell body morphology, and then from cell, and that's Layla and Forrest, and then let's bring Sharmi into the mix and zoom out and look at the dendritic morphology, which is going to come here. We actually have the synaptic input. I want to talk faster than the movie, I guess. I'm always like that. But here we zoom out to the morphology, and we're going to zoom in a second to the beautiful spine morphology that differentiates between these different types of neurons. So we have work like that. And then we have a number of studies of individual cell types, again, from this Rosetta Stone that we're using explicitly. One is just this amazing poster that Joanne gave in neuroscience that was 3 or 4 people deep for 4 hours straight, one of these animated posters where the entire astrocyte crowd in astrocyte land of neuroscience, they saw this just brand new 3D and heterogeneous look at different types of microglia and their relationships. Some of them in the previous slide, they're glued onto the cell body of a pyramidal cell. Others, only the processes go onto the pyramidal cell body, and there are these amazing relationships. There's a little cilium there, which I'll tell you about later. The amazing relationships between axons on the cell body and these microglia, which people think are involved with plasticity. And then another example, again, involved in plasticity, another type of microglia that is glued onto an apical dendrite and also processes onto the apical dendrite. another example of a study that Parastu and Jack are doing with Forrest to look at the morphology of neuromodulatory axons. Impossible needle in a haystack, but if with the database you can plot boutons per millimeter versus synapses per millimeter. In the lower right-hand corner, you see lots of boutons, but very few synapses, and those turn out to be the neuromodulatory axons that they're able to study wholesale. We have another study with Claire and Stacy on the morphology of Martinati cells. We have a fantastic study in collaboration with Caroline Ott and Jennifer Lippincott-Schwartz on some people know that many neurons have one cilium, but they went to all the neurons in this volume. Every single one has a cilium, as you see on the top, classic cell biology. And there it is, and you can study if you want the CAP version of cilia in cortex, we got it, or they got it. Okay, I'm getting enthusiastic. What about humans? We've been talking, we've always worked in mouse, and we've been talking with Ed and the team for a long time about doing cell types and anatomy in humans. I would love to talk about the early work. They cut a series of human tissue over heroically. It was like after over drinks. We'll talk about it. Cutting a human block, but I won't talk about that today. But I do want to talk about, again, my back burner project that I think is ready for prime time and ready to really bring into the fold, which is addressing, I think, and I think many people think, is the biggest gap in our knowledge about the human brain, which is the large-scale wiring diagram of the human brain. And I'll tell you what I mean by that and why it's a lack, and just the wiring, the projectome itself, but also in the context of cell types. I think it's going to be very exciting in the coming years. So the last, I'm actually doing okay for time. Good, hi. The last, I'd like to talk about whole brain studies. I am not going to talk about the whole mouse brain. We could, it's a big movement. I and others are trying to convince the DOE and NIH that it's time to do that, and I think it is. But let me talk about a different kind of whole brain connectomics that one can do in both human and the mouse and other species. Francis Crick, again, wrote this wonderful paper in 1993, which I really do remember reading and thinking about forever until we started thinking here about doing this kind of work in the human brain. And it was an impassioned commentary in Nature, written with Ted Jones, the late Ted Jones, who's one of the great anatomists of the 20th century. And the impassioned plea was clearly what is needed for a modern human brain anatomy is the introduction of some radically new techniques. But unless there's a general awareness of the need for them, they're not likely to arise. So that was the awareness of them, sort of a call in 1993. And why did they write this paper? Because it was right about the time that Van Essen and colleagues made this amazing, really first detailed wiring diagram of a largely cortical network in the macaque visual system. They introduced this study in the paper, and then they say, what does the human equivalent of the connectional map look like? The shameful answer, I wish you could write like this and get it published. The shameful answer is that we do not have such a detailed map because, for obvious reasons, most experimental methods used on the macaque brain cannot be used in humans. And I think they say it, but I'll say it. In all of the literature on the human brain, never has one single axon been traced from source to target. Not one axon. All of the connectional stuff is from lesion studies and from MRI imaging. So yes, there has been a great science of MRI imaging. The current state of the art of whole brain connectivity comes from diffusion imaging, which gives maps that correlate with, again, sort of the low resolution but informative MRI bold signal physiology. But why is it not really a wiring diagram? It's a hint, but it isn't really a wiring diagram because it strings together at each position in space, roughly a millimeter, now it's a ½ millimeter in space. There's a vector of the dominant direction that axons point, and maybe another vector where there's a subdominant direction. And these wires are not wires, it's linking together the dominant directions. Another slide, how much better can we do? What I propose to do, and I guess we're doing it now over the past several months, or year, but really in the past several months, is go from these huge voxels that have tens of thousands of axons in them, and to this. This is actually 1/6 of a millimeter, or 150 micron square, going up and down in a 50 micron. So this is 136 times 1/20th, 1/720th of a cubic millimeter. So in every cubic millimeter, there are these amazing wires going in all sorts of different directions. And these, so before I get too wild, let me say what it actually is. We would like to do an axonal connectome or a projectome of the human brain, or now we just got the first grant in this to do it in the macaque, but we're also doing work in the mouse and human. It's a light microscopy study of antibody-labeled axons. It yields a dense projectome. Everything we know about projection patterns really are from tracer studies, like the fabulous work of our friends here who do the Allen Connectivity Atlas through tracer injections. But rather than a region-to-region plus cell type map, this is every single axon that one can trace if the signal to noise is good, and it's actually very good. In the human brain, that's more than 10 billion axons, a little bit more than 100,000 kilometers of axons. In fact, the individual steps are not difficult. It's really standard microscopy, but the scale is immense. And really the only way to do it is in the modern age with better microscopes, but really using the tools, the computational tools from EM Connectomics. And it's not your textbook, what's going on in the white matter. Here's some data we put together for the grant a year ago, Emily and Rylan and myself for the first grant. My animation is messed up. In some places, the axons are highly structured and fasciculated, like the textbooks. In some places in the middle, it's partially structured. You can see what's going on, and there seem to be pathways. Most of the places, it's chaos. It's a wild jungle with 3 or 4 different directions. And the question is, which are those axons? But the beautiful thing is that it's highly traceable, that with spacing between the axons from myelin and other things, it's highly traceable, both, it's highly traceable, enough said. So what is this project? 100,000 kilometers of myelinated axons. We'd like to map the largest 50% of these, which are the ones roughly a micron in diameter, money, hopefully NIH money permitting. It has potential to provide, really, I think, the first modern histological and connectological analysis of cortical areas. It has clinical, I think, actually quite important clinical implications for a number of reasons, but one of the major is that deep brain stimulation almost certainly works by stimulating axons, and we just don't know where they all go. Secondary, but really primary, it will give a complete cell count for the human brain. Potentially, but now I think beyond potentially, along with antibody staining for a broad cell classes such as excitatory, inhibitory. And I keep adding to this sentence, just the grand challenge is correlated morphology and MFISH, and talking with Rusty and looking at his poster yesterday, I think this is possible. I think this can be combined with MFISH to give not only the axons, but their genetic identity. Just 2 more slides, actually. The preliminary data and the grant are in the macaque, but with help with expansion microscopy and processing from Reid at MIT, who's one cross fingers public statement. I hope he comes here to work on this. And again, Emily Tershak, a wonderful, wonderful RA. We processed some human tissue, and this is a bit of a gyrus of human tissue. You see a tiny square there of 150 micron region in the white matter, and this is the first example of tracing the white matter in the human with a bit of expansion microscopy. We also did this with our new fancy microscope with Kevin Takasaki and again, Rusty. But it's just as easily traced, and this is the first time out of the box, out of the freezer in the basement, not optimal tissue. And then finally, last slide. Initial studies, we tried doing this first in the mouse, and I said, it'll never happen. The mouse, this is very small now, but the mouse white matter is incredibly thin, and unlike in gyrencephalic big brains like ours in the macaque, it's highly fasciculated, and one just lost those axons as they went into the white matter. They just bundled together. I have a minute to say this, that the classic thing you hear from Max Cowan in the 50s, 60s, 70s, whenever, is the gain in brain is mainly in the stain, which is another way of saying what was the revolution that allowed us to see anatomy. It was the Golgi revolution, being able to see some cells, because if you see everything, you see nothing. And that's it, because everything interferes. And that was the case in the mouse white matter. But as we saw with other white matter, if you can get enough resolution, you can actually begin to discern axons. And with expansion microscopy, and this is really the last piece of this entire puzzle, I think, to bring this kind of work to the mouse. With expansion microscopy, this is 3 times expanded. You can begin to see these tight axon bundles in the mouse. I apologize that I finished my talk late. Thank you, Ashley and everyone else. But this is data that Emily and I collected on Saturday. And with expansion microscopy, we can have 40 nanometer pixels. And this is at the white matter layer 6 boundary. It's only 50 by 50 microns, 1,000, tiny pixels. We can trace all of those axons that are previously invisible. And the final challenge, I looked at the striatum and said, there's no way we're going to be able to trace those bundled axons in the striatum. And that's the last thing we did at 5 o'clock on Saturday afternoon. We looked at a bundle of axons in the striatum. And I don't think anyone's seen this before. That we know that there are motor axons there. I got this from, thank you, Julie. Motor axons I knew, but not corticothalamic and thalamostriatal axons. The motor axons are the big ones with the good axon staining, but the empty myelin tubes, less than 500 nanometers, really small axons, but you can trace them all. Those are the other fine ones. So I really think that we can do a dense projectome in the mouse in the context, thank you, Rusty, of MFISH. So let me conclude with the big questions again. What are the big questions? Morphological cell types, cell biology, topographic specificity, cell type specificity in the center, the most important thing. Functional specificity, didn't talk about it much, but really that is one of the major goals. And then all of these morphological categories. we want to do it, but we're actually doing it. And you've seen it over the past several days. This is my favorite slide. I just finished it. Morphology. Morphological cell types. We have 5 studies going on. We have the chandelier cells, the somatostatin cells from Claire and Stacy, the nuclei, the pyramidal cell dendrites and axons, the neuromodulatory axons, and this sort of back burner all category, which, so what's this good for? It's good for perhaps doing CAP answers to just all these individual classic questions. But there's also the all category. What is the matrix? What does the W alpha beta look like? And there's cell biology. Who thought that would be sort of perhaps the last word on cilia in neurons in the cortex? Connectomics, the determinants of connectivity, topographic specificity. That's so routine and simple in a way, and we have good models. It really becomes part of all studies by building null models of which neurons are nearby. You saw some of that in Casey's part of the talk yesterday. Cell type specificity, this huge category. Again, I'm calling out the first study that of all the inhibitory axons onto various targets, but this is a huge category that we're really working through. And then there's morphoconnectomics, which is, for lack of a better word, anatomics, whatever. Synaptic weight, the first question people ever ask is, what is synaptic weight? There are classic measures of that, such as spine volume and postsynaptic density area. There's a lovely study by Sven Darkenwald and the gang here with Princeton on that really feeds into learning rules and computation. It's a really exciting study about synaptic weight. And really a wonderful thing is we're writing a grant right now with Kristen Harris, who's wrote the classic papers on the relationship between synaptic weight and the morphology under EM to calibrate all these things, both the synaptic weight and the recent plastic history. They're morphological signs of recent plasticity. Finally, the synapse location, you saw some of that yesterday with the computational work in the pyramidal cell postsynaptic to chandelier cell inputs, but really all of MIT certainly is doing work on this currently. So that's the current set of questions, but some of them are really sort of the whole thing. What does the network look like? And then finally, the final synthesis. I don't think there's any one synthesis. Maybe a final W alpha beta will be one synthesis, but I think, as you see, it's I shouldn't say this. It's like shooting fish in a barrel. It's ripe for you have a question about the neurobiology. Just what is going on morphologically, and you can just look inside this encyclopedia and ask questions from computation to cell types to connectivity to cell biology. And really, the synthesis will be tying it up with the cell types program. And we're already doing this with wireframe models of neurons. Forrest and the gang are doing that. With the Rosetta Stone of the IVSCC data. But really, I think, final bullet point, we will, at least with the light work, but I think over the next decade with the EM, have direct readouts of gene expression, cell types, CAP cell types from gene expression, and all of these morphological and network properties. And here's the list of all the great people working on this with last names too, on the EM on the left, and LM, light microscopy, axonal connectomics, a growing team on the right. Thank you, Paul, who really made this whole thing possible. Go Seahawks. And it's all beautiful. Thanks. I went over. Okay.
Mic isn't working, is it? How do you envision MFISH and EM merging, either at the chemical level, or how is that going to work?
That was irrational exuberance. I don't know. You know, I'm feeling better and better about the Rosetta Stone, and it's not just morphology. There are all sorts of schemes you can do with nuclear morphology, et cetera. It's not going to be a dense probing of RNAs, but there are essentially barcode approaches to having nanoparticles that can be attached to probes. You know, I won't even flail beyond that. You know, it is a major goal of the field. It'll be great to have. I'd be surprised if we didn't have it in the next 15 years, but I'm not certain that we need it.
Thanks, Clay. That was really inspiring. I'm curious, though, about how your vision for mapping the W alpha beta, the connectivity matrix, sort of gels with these ideas of plasticity that might be changing that connectivity over short timescales.
Well, a lot of ways to answer that. What we're seeing, you know, absent doing a real plastic perturbation, you know, whisker trimming or something, we're seeing a steady state. So I think the statistics are going to be the statistics that exist in the steady state. Did we talk about I don't think we did talk yesterday about the extraordinarily plastic-looking chandelier cell axons. You can see growth cones and all sorts of fascinating things in chandelier cell axons. It's sort of a fantastic new story. So I'm quite hopeful that there will be morphological signs of ongoing plasticity. And another version of that is, I don't really have time to go into the details, but there's increasing thinking about looking at the final state of cortical networks as a very tight constraint on the learning rules that went into that. And actually the story about synaptic weights from Princeton is exactly that kind of story, that, you know, rather than a continuum of weights, there are two sort of quantal weights, and that fits into certain models of plasticity and learning. So I think it's a window. I actually more and more believe this is a window into development and plasticity. But very importantly, another talk is EM, not as doing these infinite volumes, but doing smaller, modest volumes as an assay. That if we can do infinitely big volumes in a couple of months, we can do a small volume a week, and that will allow people to do plasticity studies as they're doing in this grant that I was talking about and looking at the connectome. So.
No other questions? And let's thank Clay again.
Thanks.
Okay.